Great, thank you, Joe, and thank you for inviting us to present today. We're happy to be here. So Wes and I, for several years now, have been conducting research on Starry Stonewort, various facets of it, and we're gonna run through quite a bit of that today. So it'll be sort of a scan of the various research questions we've been addressing. So Starry Stonewort landed in our laps back in summer 2015. That's when I moved to Minnesota to start my position at the University of Minnesota, and with a goal of starting, among other things, an extension program in aquatic invasive species and doing research on aquatic invasive plants. And so Starry Stonewort was found in the First Lake in Minnesota right after I moved here, and so became something that we really wanted to learn more about, and became a focal area of research in my lab. I like to start out when I present on Starry Stonewort just describing exactly what we're talking about, because I think the term algae is kind of problematic because it's really an ecological catch-all rather than really a evolutionary term describing a particular group of species. So Starry Stonewort is a green algae, and that puts it here on kind of the tree of life. And what's problematic about the term algae is that it pops up all across the tree of life in very different taxonomic groups, even in the bacteria, which is where we see the harmful blue-green algae. So I think the term algae, when you hear the Starry Stonewort is invasive algae, I think it brings a lot of baggage with it that kind of has to be unpacked. It is specifically a charophyte, a Caracean green alga, and it's within this broader group that encompasses the land plants and vascular plants and seed plants that we tend to interact with in our daily lives. So closely related to the plants. It's native to Europe and Asia, and it was first found in North America in the mid-1970s. An interesting fact about it is that it is dioecious, meaning that males and females are separate individuals. And so far, only males have been found in North America. It's possible that there are cryptic females that we haven't found, but at the very least, the populations here are overwhelmingly male. And that means there's not sexual reproduction and production of viable spores. Spread here is asexual or clonal, and it's with fragments and bulbils. And this is a very zoomed in image of one of these asexual reproductive structures called bulbils, and its star shape is what gives us the common name Starry Stonewort. And so, as with many aquatic invasive species, boater movement is a presumed driver of spread in this species. So when Starry Stonewort shows up in a new place, now in Michigan, it's been prevalent for a pretty long time, but it was found in Wisconsin just a year before it was found in Minnesota. And there are, it strikes fear in people. And there are headlines like this: an invasive species spreads fast, it's the start of the death of a lake. And when it was found in Minnesota, there were similar headlines sort of saying this was really disastrous and very dangerous, and especially dangerous invasive species. But then you also hear this other side of the story. Starry Stonewort is rare. It's even an endangered species in much of its native range, where it's a conservation priority in many countries, and it's considered an indicator of good water quality and appreciated for its ecosystem and food web contribution. So people are sort of subjected to these different strong perspectives that this is a terrible species or that this is a non-threatening species. And that's because we're at this sort of early stage of the invasion process. Certainly in Minnesota, but even for North America, mid-1970s wasn't that long ago compared to some other invasive species. And a curve like this is often used to describe various stages of invasion. So for Minnesota, for example, we don't have hydrilla. It has not yet been found in the state, which is good news. And then we have something like curly-leaf pondweed that's been here over a century. In the case of Starry stonewort, we're just kind of at the beginning of this curve where it's still pretty rare and localized, and we have a high degree of uncertainty of what the future will look like, how widespread it will become, and what degree of problems it will cause. So kind of one of the overarching questions we've been asking, is this going to be a native weakling, this rare endangered species that becomes an invasive bully? And there's, you know, surprisingly frequent examples of this in the invasion biology literature. For example, pictured here, Monterey pine, extremely rare in its native range while becoming invasive on multiple continents. Phragmites australis, another species we do research on, has experienced these diebacks in Europe, even as those very same European genotypes have expanded throughout North America. Or to use an animal example, house sparrows are incredibly successful as an introduced species while declining in their native range. So is this potentially what is happening with Starry stonewort? So to address this, our focus, I guess 5 years is out of date. I guess I need to update that. But we've been looking at sort of three broad themes that Wes and I will be talking about today. So spread, where is it going to end up? Impacts, what's it going to do when it gets there? And management. How can we support rapid and effective responses? So starting with spread, here is from a review paper we did a few years ago where we were looking at accumulation of new records in North America, both things that have been confirmed with voucher specimens in that solid line, as well as just wider reports in general on that dashed line. And we can see sort of a sequence of it being found in new states and provinces, and many more water bodies being recognized as having Starry stonewort. And this is sort of what that footprint looks like, roughly as we know it today. I've updated this map a couple times with a aesthetically beautiful purple filling in of counties. There are definitely some more that could be added here, but this represents fairly well like its basic footprint in the Great Lakes region. And then this is what we've seen in Minnesota. It was first found in 2015 in this these connected lakes, Coronis and Mud. And then every year since, new locations have been found. In 2016, there was this kind of flurry of discoveries in the northern part of the state. And this is what it looks like now, where we have sort of two main clusters of Starry stonewort in kind of central Minnesota and northern Minnesota. And for those of you who are familiar with the geography of the state, we now have one known population in the Twin Cities metro. So one of the things for us to try to figure out is, where is it going to end up? And this is kind of a multi-stage question. Where can it survive in the world based on climate? So what does its envelope of climate suitability look like? And then within that, which lakes might provide suitable habitat? You need more than just the right climate, you need the right water chemistry. And then which lakes can it get to? And that's a function of boater movement as well as its ability to survive overland transport to tolerate desiccation. So our first study on Starry stonewort, this was led by Luis Escobar, who was a postdoc with Nick Phelps, and Luis is now a faculty member at Virginia Tech. And we used records of its global distribution, both as a native and invasive species, to address its climate as a factor in its distribution. And we came up with a map that looks like this for its potential distribution based on climate, which you can see is much more extensive than its actual known distribution. So just looking at climate alone, it looked like this high potential for spread, but we need to incorporate habitat suitability to generate lake level predictions. So for example, the Intermountain West, there might be plenty of suitable climate, but if there aren't the suitable water bodies, it's not going to establish there. So our next effort along these lines was led by Dr. Ranjan Muthukrishnan, who was a postdoc in my lab and is now a research assistant professor at Boston University. And we collaborated with some folks at New York Botanical Garden who are Caraceae experts who've been studying starry stonewort in the Northeast. And we looked at the chemistry of starry stonewort lakes and saw this general pattern based on their research in New York State, as well as literature from its native range in Europe of starry stonewort occurrences generally being associated with higher pH and conductivity and pretty wide trophic state ranges in terms of nitrogen and phosphorus. So we used New York to sort of train our models. We looked at what lakes is it found in now. We examined the environmental conditions of these lakes, and then we used some machine learning methods to predict where this species might find suitable habitat in Minnesota and Wisconsin. Robin Sleeth, who did his PhD with Ken Carroll at New York Botanical Garden, did some incredible sampling. These are all the water bodies. He sampled the Caraceae in New York State, and then he later basically did the same thing for New England. And those red dots are where Robin found starry stonewort in his searches. And so we focused in on this area sort of running through the center of New York State as an area where starry stonewort had been known to occur for a while and had maybe had some opportunities to sort of spread around, sample different lakes, where maybe we could make some inferences about starry stonewort being present or not, having something to do with habitat suitability and not just, you know, where it had arrived first. So it had a few decades to sort of spread around and sample different lakes. We used a few different methods and found some general agreement that starry stonewort was associated with higher conductivity, so more things like calcium and magnesium, higher concentrations dissolved in the water. Some positive associations with nitrogen and phosphorus. And not surprisingly, given that sort of more productive lakes where there was a bit lower water clarity and higher chlorophyll A, a measure of primary productivity. And then we sort of mapped this onto Minnesota and Wisconsin and had this sort of index of lower to higher risk. Here are two different methods that seem to perform fairly well, and we can look at kind of the consensus high risk and see this pattern. And that's a pattern that, it's, especially for Minnesota at least, where I have a better handle on the distribution, maps on pretty well to where we actually see it in the state. And these predictions are independent of the actual occurrence data in these states. So some sort of independent evidence that this model is getting at the right things. So that's habitat suitability. Another factor is overland transport. So where is starry stonewort going to have an opportunity to get to? And that's something that's been a real active area in Nick Phelps's lab here at University of Minnesota, using boater movement data to figure out these spread networks. And then a more sort of empirical experimental approach that we use to address these questions is desiccation tolerance. So simply, can it survive if it's on, you know, somebody's uncleaned prop or trailer? So we perform these experiments using material of different size, knowing that if you had larger clumps, that's going to hold water longer and survive longer. And we also tested how long bulbils could survive out of water. And this was work that was led by Wes, who you'll hear from in the back half of this talk. So we had the starry stonewort, and we took it out of water for different lengths of time, as short as 15 minutes to as long as 5 days. And then we had negative and positive controls. So material that was wet and alive and had never been dried, as well as things that were dried down to a constant mass and definitely killed. And then we looked at the viability of this material after different periods of time out of water. For bulbils, we looked at the sprouting, which you can see here. And for the biomass, we looked at rehydration. So was this still, living cells that were able to reabsorb water, or were they totally cooked? What we found is that bulbils were viable for less than 4 hours. So we see this very steep drop off where none of the bulbils sprouted after they had been out of the water for between 2 and 4 hours. And then with the fragments and clumps, we saw this strong size effect that we hypothesized, where the smallest material quickly died, but the larger material could last for up to 2 or 3 days. So what does this mean? The implications for spread, I would say, are that reasonable effort will be sufficient to prevent spread. This isn't something like a zebra mussel veliger that's microscopic and could be hidden away and bilge water or something. This is material that if people are doing what they're expected and required to do by law, should prevent most incidents of spread. So the risk is really from non-compliance, which, of course, we know does happen, as well as conditions that retain moisture. So these carpeted bunk trailers, for example, you can imagine situations where there's a chunk of Starry Stonewort that's missed. It's sandwiched between the hull and the carpeted material, and maybe that moisture could be retained for a pretty long time, longer than what was seen in our experiment. The next thing I'm going to address is impact. So what does this do ecologically when it arrives in a new lake? This is an area that we were very interested in as ecologists, but where there was really high uncertainty when we started working on this species. Just not enough research, but anecdotal reports of severe impacts that we wanted to take a closer look at. For example, when we wrote this literature review, there were over 200 papers on Starry Stonewort, but only 11 of them had addressed it as a non-native species in North America, and only one of those had documented ecological impacts. And I just want to point out some new work coming out of Canada, some manuscripts that have come out over the last year or so that have really added to this knowledge since we did this review. So in 2017, we established permanent monitoring plots in 2 lakes, and we resampled them annually to examine rates of local or within lake spread, as well as impacts on native plant communities. And we call that time sequence sampling. We're actually going out to the same places multiple years, so we can really document the change over time. And then we complemented that in 2018 with more spatially extensive sampling in 3 lakes to complement these permanent plots. And that's a space for time substitution or chronosequence approach, where you're looking at areas at different stages of invasion and trying to make some inferences about what the invasion is doing. And this work formed the basis for a master's student of mine, her thesis, Carly Wagner, who's now an AIS specialist with the Minnesota DNR. Carly used both of these types of plots to understand plant community impacts. What she found is that increases in Starry Stonewort cover, that's the x-axis of both of these plots, were associated with decreases in native macrophyte abundance, that is native aquatic plant abundance, as well as diversity, measured here as species richness. That was the results from the chronosequence. With the time sequence sampling, we were able to actually sort of see in real time, so to speak, the spread. And so here, this animation is showing the increase in Starry Stonewort abundance with the red bars, and then the decrease in species richness with the green bars. This is over a 3-year period. And you can see in this particular transect where we had scuba sampling along the length of it, we saw a real rapid increase in Starry Stonewort cover and essentially an elimination of the native aquatic plant community. So sort of a worst case scenario. That's one transect. By the way, these are all from one lake, Coronis. I'll show you results from another lake in a moment. We see a little more variability as we look across more transects, but a similar pattern of real strong increases in starry stonewort abundance and associated decreases in richness. So you can see some heterogeneity across our sampling locations, but a clear pattern coming through. We did the same thing in Moose Lake, and this is the same interval, the same 3-year period that I just showed for Lake Coronis. The pattern is not as strong here, but it's similar of, more starry stonewort and declining native species richness. And then we added one more year of sampling, because why not? So we had data from 2020, and we saw some things that looked like more of the same, like here on Coronis. But then we also saw some things that were pretty different. And in some ways, 2020 surprised us a little bit, because in some cases, we saw a sort of reduction of the starry stonewort and a recovery of native richness. So for example, if you focus in on this plot here, starry stonewort's increasing through year 3, but then in year 4, it's sort of scaling back, and we're seeing a little bit of recovery of species diversity. So that showed us that yes, plant community impacts are very real. They are linked to starry stonewort abundance, but there is also high interannual variability, and results can look very different from year to year. And what the question this leads to is, what is the resilience of these systems? Can starry stonewort have enough down years that the native plant community will sort of rebound and will end up at an equilibrium, or are we just seeing a little blip of recovery in an overall downward trend in those plant communities? That interannual variability also gets you thinking about weather conditions and seasonality. And so what about seasonal growth patterns? And how does that affect its potential impacts to native species? What is the degree of temporal overlap with native aquatic plants? Are they trying to sort of grow at the same time and going to be bumping against each other? So using these same lakes, Moose in northern Minnesota and Coronis in central Minnesota, they're a pretty good latitudinal gradient, at least by standards of a single state. We wanted to look at what these phenological patterns were. And this is showing biomass over the course of 2 years in Coronis. And then for 2018, we had data from both lakes. And we sampled throughout the year, including under the ice. And this, again, you can see is work that was led by Wes. We saw some interannual variability. So we see some differences in the shapes of these curves between 2017 and 2018, but sort of a surprising consistency between the two lakes, despite them being separated by a pretty good distance. For bulbils, which are going to be important for regrowth, there was very high spatial and temporal variability, just real noisy data. But if you squint, you can maybe sort of see this pattern of bulbal sprouting and consumption as we come spring into summer. So, the bulbils turning into plants and that bulbal bank being exhausted. And then a period of bulbal production and recharge as the starry stonewort has had time to photosynthesize, acquire carbon, and then invest in these reproductive structures for the following year. In terms of the phenology, we compared it to our common native species as well as other high priority invasives. And we found that it had a distinctly later phenology. So here we're comparing, this is starry stonewort, this filled these filled curves here. And then we have a large group of a few dozen native species in blue showing their broad abundance patterns. And then also in green, we have Chara species. So here are the cousins to Starry Stonewort. And you can see it's really shifted later into the fall in terms of when it's abundant than native species. And the same is true for other submersed invasives. So here we have curly leaf pondweed, which is kind of well known for its early growth, spiking here, but even Eurasian water milfoil also is showing much earlier growth than Starry Stonewort. So we've identified sort of this new temporal niche in Minnesota lakes where it's really doing something different than the other plants that have been there for longer. We were actually scooped back in the 1980s with a study on the Detroit River, which showed a very similar pattern here with Starry Stonewort being the solid line and native species being the dashed line. And they found even later growth into the heart of winter. So this kind of gets to a conundrum for control, and that's something Wes is going to focus on. So one of the strategies we use in aquatic plant management is trying to find a window when we can treat so that it's effective for the target species, but will have minimal collateral damage to native species. With curly leaf pondweed, we can just treat earlier, but at that time, Starry Stonewort isn't really up there. It's not there to kill yet. In the mid-season, by the time the Starry Stonewort is up and there's sufficient biomass to treat, native species are peaking, so we'd worry about non-target impacts. We could treat in the fall when it's basically just Starry Stonewort, but by that time it's already likely recharged its bulbal bank. It's accumulated its biomass, it's produced bulbils. And it's going to start senescing anyway, so it really might not add a lot of value to sort of kick it while it's going down. And now I'm going to stop presenting and turn over to Wes, who will talk about Starry Stonewort management, and then I'll return a little bit at the end for some of our extension work.
All right, thanks, Dan. I am just gonna maybe ask for a thumbs up that everyone can hear me. I believe I'm unmuted.
Looks good.
Excellent, thanks, Eric. Great, so as Dan mentioned, I'm gonna talk about management. So this last part of our focus. So can we support rapid and effective responses against Starry Stonewort? So what we know is that there's been many control attempts in US Great Lakes states over the past several decades, in Michigan in particular, and since its recent invasions in Minnesota, Wisconsin, Indiana, and other states as well. These approaches fall into two broad categories. So algaecides, which are typically copper-based contact algaecides, and physical removal, which is done by hand pulling or mechanical harvest. There's been many anecdotal reports on treatment effectiveness, but there's been few quantitative assessments of these treatments as to whether they're working or not. So this really is the million-dollar question: are current treatments working? And our lab sought to address this question in two different ways. So the first being controlled laboratory experiments. So we actually assessed Starry Stonewort in the lab with controls under experimental conditions. Are we seeing reduction in Starry Stonewort using typical treatments? And then secondly, we did a synthesis of control outcomes, so actually in lake management, to see whether these management efforts were impacting Starry Stonewort. And this effort was done across 3 states, Minnesota, Wisconsin, and Indiana. And I'm going to talk through each of these in turn here. So in regards to our laboratory experiments, we conducted two experiments, the first being on thalli, or the biomass, above ground biomass of Starry Stonewort. That's depicted in that figure up there at the top. And we also looked at the sprouting of the bulbils. And for bulbils, we place these below the sediment, as that's typically where they occur in nature. We looked at 6 different copper-based algaecides. Again, these are ones that are typically used for Starry Stonewort control. There's folks in here who are managers, they're probably familiar with some of these products. We looked at 5 different concentrations from a very low rate to the maximum legal labeled rate of 1 part per million, or 1.0 milligrams of copper per liter. And before I go into the results, I just want to explain that we expected, particularly these higher concentrations, to have pretty substantial effects on starry stoneworts, given what we know of some of these anecdotal reports and given our test conditions. Again, you can see these very small jars, and we're applying the algaecide directly to these jars. So I'll start with what we found from the phthalate, or the above ground biomass. And I got this figure here just to show what we're looking at. And what we did is we actually removed the starry stonewort from the jars after treatment and weighed them. So we have wet weight as our response. And what I've depicted up here is just what we found in our untreated controls. So a small amount, 0.6 grams of wet weight. And what I'll show here is the results for each of the different algaecides across the different concentrations that we evaluated, with the red line here being able to compare it to the controls. So what we found is not quite as much reduction in biomass as we might have expected, particularly for those lower concentrations, but you can see for some of the higher concentrations, we started to see some of those reductions. The only significant reductions, however, were at that 1 part per million copper for 2 different algaecides, so algamycin and comine. And what these actually represent are 34% and 40% reductions in biomass. So modest reductions, but maybe not as much as we might have expected. So next we looked at bulbils, and this figure is to show you what we were looking at here. So we're actually picking up each one of these jars and inspecting them for bulbil sprouting. Again, these bulbils started below the sediment, so just covered in the sediment, no more than 1 centimeter underneath. Again, we expected that there might be some protection of the bulbils from these copper contact algaecides. These algaecides need to actually make contact with the algal structure in order for them to be effective. So we weren't sure quite what we'd see, but we knew that there could be some protection there. As you can see from this figure, we did see sprouting. And so those results I'm going to show here. Here our response is the proportion of the bulbils that sprouted below the sediment. So this is just a scale from 0 to 1. In our controls, we had pretty good sprouting. So 60% of the bulbils we planted sprouted. And then here's the results for the actual treatments. So this was a little surprising. We actually saw greater sprouting or equal sprouting in all of the treatments compared to the controls. And we actually saw significantly greater sprouting for some of the lower concentrations that we evaluated, specifically 0.25 parts per million of copper. Again, this was surprising. And it seems to suggest that there may be a stimulatory response. So perhaps a small amount of the algaecide is actually getting down below the sediment and causing bulbils to sprout. This is certainly something that requires some further investigation because it is concerning if this is really what's happening out in nature. So just a quick recap of those lab experiments. So modest reduction in biomass, again, for a couple of those algaecides at the highest concentrations we evaluated, and possible stimulation of bulbils below the sediment. And so, this was one of the first laboratory trials of starry stonewort control. And so folks might see this and be a little surprised. They, might want to see it verified by other studies. And luckily enough, there's actually was a concurrent study that was done by some folks from the Army Corps of Engineers. And they found very similar reductions in biomass. So they actually used small tubs outside. So little kind of mesocosm study here. But again, very similar biomass reduction to what we observed in our study. And what they did find is they had slightly better results for their bulbils. So there was more control for bulbils. However, those bulbils were actually attached to the phthalate and floating in water. Conditions that may occur in nature, but are less realistic and less likely to be the typical situation where we find bulbils. So again, those are laboratory trials. They tell us a lot of information, but we were really interested in what was actually occurring out in the field where folks are actually doing management. So what we did is we did this synthesis of control outcomes. So we worked with the Departments of Natural Resources from Minnesota, Wisconsin, and Indiana, as well as private contractors and folks who are doing management. We worked with those folks to acquire any and all survey data from the time of initial infestation until the current time, and that's data from 2010 through 2019. And we just wanted to compile that information to be able to assess whether these treatments were actually effective. So in regards to the management data, here's what that looked like. We got information on around 400 total different management events, or total treatments, as we're calling them here. And the vast majority of those were algaecide treatments. And was pretty much what to be expected. You can also see some other types of treatments that we got data on: hand pulls, mechanical harvest, DASH is diver assisted suction harvest, and then some other less frequent treatments as well. So again, the vast majority of these are algaecide treatments. The vast majority of those are copper-based, so the kinds of algaecides that we were looking at in our experiments. And what these amount to over time in the data that we collected, it's about 20 tons of elemental copper across these 3 states since the time when the states were first infested. So in regards to the survey data that we collected, this came in 3 different types. So the first being data on whole lake point intercept surveys. So these are surveys that are done on a grid across a lake where at each point plants are collected. And we were specifically interested in at how many points across the lake was Starry Stonewort found. So for these data, we're looking at a whole lake response to treatment. We also looked at treatments that were conducted in targeted areas. So we looked at survey data from just within these targeted areas, so what we're calling targeted sub-PI surveys. And lastly, we received data on what we call case studies. So smaller scale studies that typically had an untreated reference area that we could compare to the treated reference area. Some of these studies also looked at things like biomass and also some less frequent types of treatment. And really, these represent a scale from the greatest spatial extent to the smallest, and also the most data to the least data. So we have a lot of data on these whole lake PI surveys. They're done every year and across these states. So we're able to collect a lot of data from those surveys. So what does that whole lake PI survey data look like? We have data from 35 lakes across 10 years, which amounts to a total of 153 PI surveys. And the type of data that's collected in these are 2 types. So frequency of occurrence, so that's just the number of points out of the total points where Starry Stonewort was found. So it's a proportion from 0 to 1. And then rate density, which is a measure of abundance. So one of the first things we were really interested in looking at was what's happening in these lakes over time. And so here we have Starry Stonewort frequency of occurrence as our response, and the year since infested on the bottom. Year 0 meaning that was the first year that it was infested. And here's what we found. So this may not be surprising to many folks who've studied Starry Stonewort, but a substantial and significant increase over time in the extent of the lake that's infested since the time of infestation. And these lines here, I should say, are for each individual lake. So one thing you might notice is there's this pink line here that seems to be going against the grain. And what that pink line is for Pike Lake in Wisconsin. And this is the only lake among these where we have greater than 2 years of data and where the lake was never treated. So this indicated that maybe there's something surprising here in these untreated lakes. So then when we actually analyzed the data, what we looked at was whether or not we used those surveys to see whether or not there was treatment done before a survey in a given year. So on this graph that I'm going to show, that would be if there was no treatment done before that survey, that's going to be on one side. And then if there was treatments done in a survey, before a survey, that's going to be the other side here. And again, our response here is frequency of occurrence. So are those treatments keeping the extent of Starry Stonewort within a lake contained? And what we found was that there was essentially no difference whether a lake was treated or not in the extent of Starry Stonewort across the lake. And you can see that indicated by the p-value up here, which is not significant, and those error bars that overlap substantially. And what I will say here is that this model that created this image here, we accounted for things that might confound the results. So the year since infested, survey day, the amount of lake that was treated, and we counted for each individual lake, as lakes might have their own response. So that was for a whole lake response. We know that treatments typically aren't done across a whole lake, they're done in a small area. So to hone in on the actual effect of treatments within a treated area, we looked at these sub-PI surveys. So these are done before and after treatment. So the data here is going to be shown for what the frequency of Starry Stonewort occurrences before and then after treatment. And what we found was there's actually no difference between the Starry Stonewort frequency of occurrence within these treated areas from before to after treatment. You can see those error bars substantially overlap, and the effect is not significant. Now that's for frequency of occurrence, but we also had abundance data, so that rate density data. And when we looked at that, we saw a very similar pattern. So again, no difference from before treatment to after treatment. So with those results, it's pretty clear that we don't appear to be reducing Starry Stonewort to pre-treatment levels. But what we don't know is would there be even more growth if we had not treated? So we know that as time progresses, we expect there to be more growth throughout the summer. That's what our phenology results showed. So really, in picture form, we know this. This is what our data seem to suggest, that with copper treatment, we're not seeing any less Starry Stonewort in a lake. But would there be even more if we had not treated? And really, to get at this, to get the answer to this question, we need more untreated reference areas on lakes that are monitored, and more just untreated reference lakes, so like that Pike Lake in Wisconsin. Again, such that we can compare treated results to untreated results. How this looks in terms of an experimental setup or a survey design setup is what we call before-after control impact, or BACI design, where essentially over time, we've surveyed both treated, which is the black outlined area, and untreated areas, which is the gray outlined area, from before and then after treatment. So we can compare those two treated areas. We do have some situations where we have data like this. So in particular, Lake Coronis. So I'm just going to walk quickly through this little case study. So Lake Coronis has been infested. Dan mentioned that's the first lake that was infested in Minnesota in 2015. There's been ongoing management efforts that have been both algicide and mechanical harvesting over time since infestation. The data that I'm going to show right here is from 2016. And in this situation, we have mechanical and algicide treated area, an area that's just treated with algicide, and then an area that is a reference area. There's a couple other areas on this lake, but these are the ones that we're going to be most interested in. And we tracked these areas over time and measured biomass. So I'm going to show that on this figure here. So here our response is the Starry Stonewort biomass, and note the log scale. And so again, here are our three areas. We have our two treated areas and then our reference area. And so we monitored over time in the reference area, and this is the line that it produced. So we know what's going on there. So essentially this tells us what would happen if we had not treated. And then when we look at the two treated areas, what we actually found was a really substantial and significant decline in biomass in these two treated areas. So again, one that just received an algaecide treatment, one that received an algaecide treatment and mechanical harvest. So again, this is showing us that in at least some situations, these treatments can be very effective, essentially dropping the biomass by nearly 100%. So presenting here another small case study. This is for Grand Lake. So this lake, Starry Stonewort was first found in 2017 through a systematic statewide search called Starry Trek that Dan's going to mention here at the end of the talk. Essentially what was found was a small infested area near an access. So unlike Coronis, this is a very small infestation, which was deemed very manageable. So what was done in this lake was multiple hand pulls that were done from the first time of infestation, and that there was no algaecide treatment done here. So I'm going to show the results from these treatments, and this is going to be in terms of biomass removed. So what we see is that first, in 2017, when folks first went out to remove biomass, there was a lot, so 450 grams. When they went back out, there was essentially no biomass left later in the season. And you'll note that this is September, when we'd expect Starry Stonewort to be at its peak biomass. Subsequent years, more biomass was removed, and you can see that year after year, we're seeing subsequent reductions in biomass and the amount that's found. So this is very promising results from this hand removal. But again, it should be noted that this is a very small infestation that's isolated as well. So just a brief synthesis of those outcomes. First of all, we know from those lake-wide analysis of those whole lake PI surveys that the population extent is significantly increasing over time in infested lakes. So the vast majority of those lakes were increasing over time. We also know that current treatment practices don't appear to be reducing that frequency of occurrence or that population extent. In regards to those within treated areas, we can see that both frequency of occurrence and abundance don't seem to be affected by treatment. They are largely the same. But again, we need more untreated reference areas to compare that with. Lastly, from these case studies, we can see that treatments can be effective at reducing biomass for nuisance control. That's what we found in Coronis. And we also found that physical removal, can be a very effective method of reducing biomass. So this would be, again, hand pulling for these small infestations. With that, I'm going to turn it back over to Dan. He just has a couple last slides to share with everyone about early detection search efforts.
Great, thank you, Wes. I'm back. So Wes mentioned one of these lakes that was found during an event called Starry Trek, and this is a statewide single-day volunteer search effort that we've done every year since 2017. We have 20 plus meetups around the state where local coordinators train and manage volunteer participants. So these are often county AIS staff or people with local units of government or people who have gone through our AIS extension training programs in the past. And then they send these volunteers out to go search public accesses for Starry Stonewort. And we coordinate with a sister program in Wisconsin called AIS Snapshot Day that's led by Paul Skowinski of University of Wisconsin Extension and his collaborators. And one of the things we do each year leading up to Starry Trek is spend a good deal of time figuring out where we're going to send our volunteers to search. And so we combine that risk model that I showed you earlier, where we have kind of a risk rating for each lake, as well as the expertise of our coordinators who manage the volunteers and host these meetups. And we ask them where they want to search. And they bring up things like accesses being busy, high boater traffic being important as a entry point into the boundary waters, for example. And so we kind of combine these sources of information, and then we set up routes for the volunteers to go out on. And we've been pleased to have really high and growing participation. I need to update this with 2021 results. This slide was put together by my colleague Megan Weber, who's an extension educator working on aquatic invasive species. But even with our coronavirus impacted years in 2020 and 2021, we've had really good turnout. And sort of frankly, to my surprise, it's been a pretty effective way to find new populations of Starry Stonewort. These 200 plus people are, they're volunteers. Very few of them have botanical experience or expertise in aquatic ecology. And with a pretty simple training and a protocol of doing rake tosses from a public access, they're responsible for finding about a quarter of our known populations in Minnesota, including small populations like Grand Lake that Wes mentioned, where it's enabled a rapid response and a hand pulling approach that seems to be quite effective. So I want to acknowledge our sources of funding and some of the people we've worked with on this work that Wes and I have covered. And yeah, we would be happy to take any questions you might have. I'm going to stop sharing so I can see controls better. And yeah, thank you for being here and listening to our presentation.
Man, that was wonderful. Thanks, Wes and Dan. I learned a ton. I think I can probably speak for the other folks in the audience. A lot of new information, and clearly some of this has not yet hit the streets in the peer-reviewed form. So it's nice to see that early snapshot of what you guys are producing, and really, really fascinating stuff. And I think important for management as we all tackle this problem together. So
thanks, Joe.
There are a lot of questions that I'm seeing coming in through the Q&A function. I'll just remind folks, if you have a question, please use the Q&A function. And so what you can do is hover your mouse down to the bottom of the Zoom window, you'll see a Q&A icon there. Just click on that and you can submit a question. We've got 7 thus far, and I've got a couple others collected plus my own questions. So I'll just take them from the top here. The first question is from Paul Skwinski. In the initial results you guys talked about with bulbils surviving less than 4 hours out of water, what were the humidity and air movement conditions in that experiment? Was it dry out? Was there a lot of air movement?
Yeah, so I just pulled up the paper that those results came from, and so we actually did 2 experiments, and I believe the ones that Dan presented were done inside the laboratory, but we also did a similar experiment just outside the lab. So the numbers I'm seeing right here are about 40% relative humidity inside the lab. So not terribly dry, but also not what you might find on a hot summer day or a muggy summer day. So that was that. I'd be happy to either share this paper with Paul or any other folks who are interested in some more of the details. So yeah, and I will say again that what we found actually in the outdoor experiment where we had some air movement, just natural air movement and natural sunlight, is we actually saw bulbils dry out much quicker than that 2 to 4 hour threshold that Dan presented on. So, but again, those conservative results of what you might see under more controlled conditions for that 2 to 4 hour time period.
Yeah, hi, Paul. And I would just add to that, yeah, our results are probably sort of a high end for bulbul survival in some ways, in that we didn't have, the air movement that would happen for exposed bulbils as it's, driving 60 miles per hour down the highway. And some of these studies will have fans trained on the material. So we didn't do that. So on the one hand, bulbils could survive longer if they were at the, sort of piled under wet gear. You could potentially see much longer bulbul survival. On the other hand, you could see much shorter survival if it was exposed to air movement going down the highway.
Exactly.
I'm imagining turning on a fan at a pile of bulbils and watching them all just fly off of your lab table. Another question that was sort of related. You guys talked about the sort of benefits of having a moist environment to bulbul survival. And you posited that, for example, in between a bunk and a boat, that would be an area that might retain moisture for quite some time. But also it would be dark. And so do you guys have any anecdotes or evidence about the effect of dark versus light on bulbul survival? Do you think that darkness might increase mortality in those shaded, compacted areas?
I'll take a quick stab, Wes, and then if you want to jump in too. I don't think it would, because where you see the bulk of the bulbils is very shallow in the lake bottom. So it forms, Starry Stonewort doesn't have roots, but it has what are called rhizoids. It looks like fishing line, and it's just attached right at the sediment surface. And that's where the bulk of the bulbul formation is. And they'll be sort of nestled into the sediment. So I don't think that lack of light could maybe even be beneficial in terms of how long they survive, would be my guess.
Yeah, and I would reiterate that. It's not something, a lot of these questions, it's like, oh yeah, that'd be another great experiment to really test that. But yeah, I would echo Dan's statement and say that I think really what the bulbils, when they're underneath the sediment, yeah, they're starved of light. They don't have light. It's like a seed essentially. But once it gets enough heat, is typically what we see is then it's going to start sprouting. So we can keep them refrigerated, at 4 degrees C, and they're fine. But as soon as we give them some warmth, they'll want to start sprouting. So I doubt that darkness would prohibit sprouting in any way.
And what would your answer be for kind of an analogous question for the Algae? Is that the word I'm looking for?
Yeah, those are certainly photosynthetic materials. So, I think for that, probably it's probably tough to say. I think that, being sheltered in that area, it's just more of moisture retention. Really that material, they're pretty delicate. So actually it could get crushed in that situation if it's, pushed between 2 surfaces, and that can destroy the material. Unlike some, milfoil stem might be more strong or resistant to something like that. So Starry Stonewort's pretty fragile. So that's something to consider in that situation. But yeah, I don't think the darkness per se would have a substantial effect on its survival, even that thalli.
Great, thanks. Yeah, I'm imagining my identifying characteristic for Starry Stonewort of popping.
Precisely, yeah.
Satisfying. Okay, for the time sequence sampling, were there specific natives or habitat types that were more resilient to becoming a Starry Stonewort monoculture?
Yes, that's a great question. Carly, my master's student, did look at sort of species-specific responses, and she also categorized all of the plants in her data set into sort of 8 different functional groups based on their kind of architecture, growth form, whether or not they were rooted. The species that were most sensitive were what we called the carpet-forming species. Those were naiads and chara species that were essentially trying to do the same thing that Starry Stonewort does. So, physically in terms of the growth form, sort of occupying that same niche of spreading on the bottom and forming those dense beds. Those were the most sensitive species. And then conversely, some of the kind of floating leaved and taller statured species that were just headed right to the surface were less sensitive, presumably because they're just kind of escaping from that competition.
That makes sense. Okay. During the study, when you measured the change in Starry Stonewort versus natives over the 3 years, and I think it was 4 years in the end, were any of those lakes trying any treatment methods while you were studying those plant populations that might have confounded the results?
Yeah, good question. Yes. So there were, particularly on Coronis, they've had an active treatment campaign multi-year. So what we did is we coordinated with folks in the DNR and the Lake Association to determine where they would be treating, and then did our sampling far enough away that we felt like it wouldn't be impacted. There were, I think and maybe I'm mixing up with the phenology data, but I think there was a case where one of our untreated reference areas ended up getting treated, and then we pulled it out of the analysis. But I feel fairly confident that it was far enough away, hundreds plus meters, that it wasn't being affected by that copper treatment.
Great, thanks. What is more effective, treatment then physical removal, or physical removal then treatment? So it's a question of sequencing.
Yeah, I mean, what we found on that study on Lake Coronis was an initial harvesting, and then treatment actually seemed to be effective. I hesitate a little bit to make a recommendation based on that because it is very case specific. That was just, as I mentioned, it was one case study. So where there's a bunch of biomass, and maybe I believe in that situation, it was still a little bit of a haircut with mechanical harvest. So there was still quite a bit of biomass there. That subsequent algaecide treatment was effective, but there may be situations where that's not going to be the case. So, and we also know, mechanical harvesters, there can be issues with spreading Starry Stonewort or spreading, any aquatic invasive plant. So certainly Starry Stonewort can fragment very easily. Those bulbils can get uprooted and float away. So that's something to, strongly consider if you're thinking about using a mechanical harvester as well.
Okay, thanks. And I'm just going to check in here. I do have that it's just a little bit past the top of the hour. And we've got 10 questions remaining. So if you guys are okay with it, I'll ask a few more questions. But I do want to, understanding that some folks might have to hop off or might have left us already, but for the remaining folks, you're more than welcome to hang on and listen as we answer the questions. Appreciate you attending. And of course, we will follow up with an email with a video recording of this webinar and the contact information for Dan and Wes. So if you have questions, you can follow up and ask them follow-up questions as well. Obviously, a lot of interest and a lot of questions here. So, I'll keep going. Let's see. So just a clarification question. Using low concentrations of copper may stimulate bulbils to grow. Using copper or copper with removal methods generally can reduce biomass. Is that?
Yeah, that's another, it's a little bit complicated, but certainly our laboratory trials showed that that possibly could happen with bulbils with copper treatment. And we saw that actually, I didn't present results from Coronis where we saw a similar result in the field. So that's, I think that's something that requires a little bit more research, but it's been now this lab study and we've seen it in the field. So it's, you know, we've gotten a little bit of evidence on that. In terms of bulbils' response to, you know, any other above ground treatment, I don't think we really have a great handle on that. So again, on Coronis, even after, you know, there's above ground algaecide treatment, most of the biomass was removed, we actually still saw bulbils sprouting and potentially more bulbil density. So more bulbils actually after that. So that bulbil aspect is still a little bit tricky. So I don't think I'd have a, you know, really a good recommendation for how to deal with those bulbils in the sediment. And maybe Dan wants to jump in on that, but I think that's my take right now from the information we know.
Yeah, I agree. I think I know there's a lot of interest in developing chemistries and methods that could get at that bulbil bank. I think at this point, sort of the optimistic view would be that you could have effective enough treatment of the biomass that you could sort of exhaust those bulbils over time, you know, knock back the biomass, they sprout, turn into plants, you knock them back again. But I think, yeah, we're not at the point where we could have any confidence of directly getting to the bulbils.
Yeah, and I'll just add one more thing onto that. And I think this gets back to that, you know, kind of when to treat, you know, issue as Dan mentioned, that certainly later in the season, there's a lot of bulbils that have already been produced and you see this early in the season. So really that maybe that August, September time could be beneficial to treat, but you also have to be careful of those natives. And what we've seen is in mixed beds of Starry, Stonewort, and Chara, you know, when treating with these copper algaecides, you'll see off-target effects on those other native Chara plants. So, you know, if you have a pure bed of Starry, you know, there's probably an ideal time, midsummer, mid to late summer, that you could treat and use that approach that Dan mentioned, which you maybe could exhaust that the resources before when bulbils are produced.
Okay, thanks. Yeah, I think sort of have a class of questions where people are wondering how effective are other approaches, whether those are other herbicides, not just copper, but the other typical aquatic plant herbicides. How effective are harvesters and some mechanical harvesters, and then also DASH. You know, so I guess more of a general question. Do you guys have any sense for the efficacy of those treatments as opposed to copper?
Yeah, I'll take a stab, and then if you want to hop in, Wes. The one treatment approach that I feel very good about, but it's limited in its application, is hand pulling, because I think we've seen very high effectiveness. It requires persistent effort. It's not a one-time thing. It's, you know, year after year. But at Grand Lake, we have seen sort of a containment to that small initial area that was found and steady reductions in biomass. So, but yeah, how scalable is that? You know, you reach a size threshold where it's just not practical. I am really eager to see more evaluation of DASH. What's Brad? If somebody remembers Bradley from Wisconsin's last name, he was a county AIS specialist who oversaw some ambitious DASH efforts on Silver Lake in Wisconsin. And I think, you know, they weren't able to eradicate it, but I think, thank you, Brad Stecker, thanks, Paul. You know, they really knocked it back. And so I think DASH holds some promise for those maybe intermediate scales where it's too big for a hand pulling, but it is something you could have a diver swim through. There was a presentation by the Starry Stonewort Collaborative showing some DASH treatments on one of the Finger Lakes in upstate New York. Again, you know, what I saw was just like a very impressive video. It wasn't, you know, monitoring data, but I think there's some hope there, some promise there, and I would love to see evaluation. As Wes showed, DASH just sort of barely popped up in this data set. And so it'd be great to have more monitoring data there. I lost my train of thought, so I'll pass the baton to Wes to carry on.
Yeah, thanks. I think that sums up my thoughts as well. I think I could understand how, some of the results might be concerning about these copper-based treatments. And I think it's really more of just managing expectations, knowing that in some situations, these can be effective, as we saw in Coronis, but across the board, they might not be a catch-all. And, we also know about these off-target effects. So just knowing what it means when you use that kind of treatment, I think is important. So it's not to say that those are never going to work. It's just, knowing there might be situations where you might expect it not to, and other ones where you might. So there's other kind of nuances. I know we've heard lots of ways that people use copper, whether that's, cooling it down or sinking it to the bottom, different things that may or may not be effective. We just don't, unfortunately, have enough data on those to really know if those are really effective or not. So more evaluation, that's why we kind of make that plug for, have some untreated areas and monitor those alongside those treated areas. That's really going to get us what we want to know, as folks who are interested in managing Starry Stonewort.
I just want to jump back in to emphasize a few points. I think that what Wes said about expectation setting is really key. And so I think at least what we know so far and what we've tried so far, the expectation should be set at the ability to have nuisance reduction and not at this is how we're going to contain Starry Stonewort within a lake, which we can do with your Asian water milfoil and curly leaf pondweed, is actually, knock back that lake-wide extent. And then, yeah, you mentioned, combining with herbicides. A lot of the treatments now do combine copper-based algaecides with herbicides. That's not something we tested in the lab. We do have it in our synthesis data set, but the Army Corps folks, that report that Wes showed some of the results from, they did do combined treatments, and really it didn't look in that lab setting at least much better than what we saw with the algaecide. And then lastly, and then I'll stop, just want to climb up on my soapbox for a moment and say that we really are flying blind because of the lack of monitoring of untreated reference areas. And if we had, Wes highlighted that one lake in Wisconsin, Pike Lake. If we had 10 Pike Lakes across the Midwest, we would be able to draw much stronger, more confident conclusions about effectiveness of treatment.
Yeah, and there's about 40,000 lakes in the Midwest Glacial Lakes Partnership. So, yeah, the ability to take a small area on 9 or 10 of those lakes and use it to inform management of 40,000 lakes. You think about the benefits would be really huge.
Absolutely.
I'll tag on to your plug, your soapbox. We have a couple other questions that I think I'll kind of lump together. The first being, okay, so if you're out on your boat and you encounter Starry Stonewort, or maybe you encounter it when you take your boat out of the water, what should you do with that Starry Stonewort? How should you dispose of it properly? And then the second question is, when you're moving your boat from one water body to the other, what measures should you take to make sure that you minimize the spread of Starry?
Yeah, I think, first of all, it's always just, if there are certain laws in place, follow what the laws and regulations are, but certainly just standard boat cleaning procedures, as Dan alluded to with the desiccation study, can be pretty effective. Walking on your boat, making sure there's no plants hanging off. One place that we've seen, again, this is anecdotally, but Starry Stonewort occurring is in anchors. So when you drop an anchor into a Starry Stonewort bed, baubles love getting into the little crevices in your anchor. And, that's also true of other aquatic plants, curly pondweed, turions, and other things too. So that's a place I would highly recommend anyone check if you've been on a Starry Stonewort lake. Scoop that mud out and make sure you're checking that. And in terms of disposal, again, if there's local regulations or things in place for how to dispose of that, but otherwise we know it dries up pretty quickly. The standard procedure that I see is truck those plants on the pavement and let them dry out. But again, and we know again from Starry Stonewort, those desiccation experiments, that it should dry out pretty quickly. So that's what I would say to do. And again, one thing as Dan showed too, with those larger clumps of Starry Stonewort that can remain viable for a while, those should be pretty easy to spot if you're doing a good check of your boat. So getting those out of there should really minimize the spread of Starry Stonewort.
Thanks, guys. A couple questions now about Michigan. So, folks noticed that you didn't have Michigan. Obviously, I'm from Michigan. It's a whole special place in my heart. Is there a reason why you haven't been doing studies in Michigan, and do you have any plans to work in Michigan?
Yeah,
plenty of Starry.
Yeah, good question. I did feel a little bit self-conscious about that. One of the reasons, frankly, is because you guys have had so many more Starry Stone. You've had Starry Stonewort for a lot longer and many more Starry Stonewort lakes. And so I think early on with, it being found in Wisconsin in 2014, and I want to say Indiana in 2012, those states were, there was sort of more consistency in really knowing exactly where the Starry Stonewort was, because it was so new. It set off alarm bells. People sent samples to Ken Carroll, voucher specimens for confirmed identifications. Whereas Michigan, by the time we started working on this, there were over 100 lakes, maybe 200 lakes. And as happens when an invasive species becomes older, it's tracked less sort of rigorously. So I think that was a big part of it. And then also just proximity and relationships, working with Paul Skowinski, for example, as a neighboring state. And then also for the synthesis project, we had pretty standardized data collection because there were point intercept survey data associated with all these treatments, which I could be wrong, but my understanding is that wouldn't be the case with Michigan, or at least not necessarily.
That's correct.
That plus just a bias against Michigan and Michiganders.
Being the coordinator of the Midwest Glacial Lakes Partnership, I often find myself wading into the Wisconsin versus Minnesota debate. I didn't think I was going to get into a Michigan debate today. Okay, last question. This is a question on resistance to chemical algaecides. Have you seen any examples of Starry becoming resistant to algaecides?
Yeah, and as I say, nothing that I'm aware of, or that I'm speaking for both Dan and I, that I'm aware of that would occur. I mean, most of these are, well, the copper-based algaecides are they're contact-based, and I'm not super well versed in all the literature, but again, we haven't seen anything like that for Starry Stonewort where it's become resistant or anything like that. So I think it's really what it comes down to is just these algaecides have been used for things like filamentous algae or algae that's less robust than Starry Stonewort. And I think it's kind of, it has it's a physical resistance, if anything, to those algaecides. And again, I haven't seen anything about it increasing resistance or tolerance over time or anything like that.
Yeah, and I'll say, first of all, a caveat that I don't know of anybody looking for it. So take it with a grain of salt when we say there isn't any, but I think there's less kind of grist for the mill for evolution of resistance because it's a clonal species. There's very little genetic diversity across the landscape, and you're not going to have those kinds of classic natural selection effects where you'll have those mutations arising during sexual reproduction and then kind of those, surviving little nodes of resistant genotypes. Another, when you said resistance, I kind of went to like an ecological sense. And I think that's something, we'll probably never be able to know, but I did hear anecdotally, and I'm curious, if you have any thoughts on this, that some of the lakes where Starry Stonewort was found in Michigan were, some of the lakes that had pretty aggressive plant management campaigns. And that could just be because those are big popular lakes. And so they get AIS. So there's been AIS treatments, but also, one thing we don't know about is, Starry Stonewort, I think of as an opportunist and kind of a pioneer species that can spread quickly through open areas. We see that in its association with accesses where there's, prop wash and bare areas. So, could that be one factor in where it's been able to really kind of establish like I said, we'll probably never know, but there could be some kind of loss of biotic resistance that it took advantage of.
Yeah, I'm not aware of any, you know, I think it's pretty hard to parse apart that correlation between the heavy use and the existence of Starry and other treatment regimes going on. So I'm not aware of anybody that's tried to disentangle that pattern and could point to one or the other or both interacting and increasing Starry. We do have concerns, you know, within Michigan of, and I think it's probably everywhere, as you mentioned, the treatment of Cara. And when you treat Cara and you leave a substrate open, that's prime habitat for Starry to come back in.
Yeah.
Given how ecologically similar they are, that's a concern. So not an invasive, at least in Michigan, but still a treatment concern.
Yeah.
That's, yeah.
Go ahead.
I was going to say, I think that is, I think I mentioned that before too, just how, you know, we know that they kind of had the similar habitats and kind of compete for some of the same areas. So there, yeah, certainly is this concern if you're treating both, is Starry Stonewort maybe coming out as the winner in some of these areas where they're both getting knocked back. And that's definitely not what we want in those areas. Yeah.
Yeah. I do have one additional question. So there was an initial flurry of findings that you guys found when you were of Starry Stonewort presence and absences that you found. And the question was that based on an active search for the species by a department or group because of incidental reports from citizens, or how did those initial findings, what was the source for most of those?
Yeah, I think it was a flurry of kind of pent up identification, so to speak. So I should say Coronis, when it was discovered there in 2015, there were like 200 acres of it. So it did not get there in 2015 or even 2014 or 2013. It had been there for some unknown period of time to get to that extent. And, you know, that cluster in northern Minnesota that was found in 2016 was largely by one AIS specialist up there who, you know, Starry Stonewort was found. There was a lot of discussion and concern about it. And then he went back out to these places where he had seen, you know, funny Cara. So I think a lot of that initial flurry was sort of the low hanging fruit of the populations that had already been there. And now people were aware of it and looking for it. And the $1 million question is, how much is there actually? Because our search, you know, it's not an easy species to find necessarily. We have imperfect detection. So the lakes, we can be very confident that the lakes we know of as having Starry Stonewort are just a subset of the actual infestations. And the question is, how large a proportion of the actual infestations are they?
Yeah. Okay. We have gone through all of the questions. I appreciate you guys staying over 23 minutes long, as well as the folks that have hung on and participation. So thank you. At this point, you know, if we were doing a live presentation, we'd all be clapping for you and cheering you. So allow me to say thank you on behalf of the participants. Really appreciate it. To the participants, be sure to check out the next webinars in our series. Next up on March 22nd, we've got one on best management practices for watershed protection, including a really neat program to reach out to folks that are just moving into an area. And then we've got other work on shoreline restoration and outreach, tribal adaptation menu, and wake boat waves later in the spring. So a lot of interesting stuff to check out this spring. With that, I will wrap us up. Thank you again, and I hope to see you all soon.
Great. Thanks, everyone.
Thanks, everyone.